Assalamualaikum warahmatullahi wabarakatuh. Today we will present about the journal that we browse from internet. It is about photosynthesis and respiration. But before that, I will introduce my team. My name is Rusdatul Isma and my partner is Nava Sayyidati Akmalia. So from the journal that we learn, we will talk about the introduction. It is about the photosynthesis and the respiration, material and method, experiment and result, and the last is discussion of result. And it is this part is include of the conclusion. So photosynthesis and respiration are the fundamental processes in the production and consumption of dry matter. These processes will be analyzed in order to gain a clue to the favorable effect of the low temperature. For the experiment and photosynthesis, there are material that needed for that. The first is plexiglass container, 5 liters. The second is a fan. It will be used inside of each container, provided sufficient mixing of the air. And the third is respirometer to determine the rate of respiration of the leaves. So based on the journal, the experiment were conducted with else grade, and the rates of photosynthesis and respiration in small plants enclosed in plexiglass container. So for experiment and the result, the first is intact plants. In this part, the plants were subjected to the following treatments. The first, constant 26 degrees Celsius and continuous light. The second, constant 26 degrees Celsius and 16 hours of light followed by X hours of darkness. The third, constant 7 degrees Celsius and continuous light. And the fourth, 16 hours at 26 degrees Celsius followed by X hours at 7 degrees Celsius and continuous light. So we can see from this figure, the net photosynthesis and the respiration were recorded continuously for 7 days. This figure illustrates the carbon dioxide gas exchange during the first and the 7th day of the treatment. The figure shows that the net photosynthesis start for all treatment at 26 degrees Celsius the first day and decrease gradually at 7 degrees Celsius and reach a fairly steady rate. The plants receiving continuous light and constant 26 degrees Celsius showed injury after 7 days, but there was no significant decrease in the rate of the photosynthesis, and the rate of respiration measured afterward in the same plants at 7 degrees, at the same plants at 26 degrees Celsius. So for the next slide, that will be explained by my partner, Nava Sayyidati Akmalia. For Nava Sayyidati Akmalia, time is yours.
Okay, thank you for Rusdatul Isma for your explanation. The presenting will be continued by me. Okay, do you know leaf tissue? Leaf tissue is a collection of the cells that have same function and structure that would form leaves. Okay, the next experiment is about respiration in the leaf tissues. Leaf tissues. The purpose of the next experiment was to find out if there are any detectable diurnal fluctuation in the rate of respiration of tomato leaves and established for curtain tissues. Next. Okay, and in this picture, okay, we know that mature leaves have faster respiration than young leaves and old leaves. In this picture, when to the rate of respiration in millimeter of oxygen taken up per hours per leaf for young mature and old leaves. Next, the last at a discussion of the result, the net photosynthesis and dry matter have an optimum temperature. When the temperature was contains, the optimal result I'm sorry, the optimal temperature was higher than 7 degrees Celsius. There are also indications that the rate of respiration in continuous light at constant high temperature might increase because of water device. And the experiment on the respiration of the leaf tissues showed that young and mature tomato leaves respiration at a very high rate when compared with other tissues. Okay, thank you for your attention. I'm sorry for our mistakes. Wassalamualaikum warahmatullahi wabarakatuh.